On a rocky ledge of Mount Carmel in today's Israel, a small community laid its dead to rest some 140,000 years ago and left behind a mystery that still pulls us in. Unearthed in 1928, Skhul Cave yielded the remains of more than a dozen individuals. The first near-complete skeleton was a child of about 3 to 5 years, probably a girl, whose bones show an arresting blend of modern human and Neanderthal traits. New micro-CT scans sharpen that picture and fuel a bold idea. This may be the earliest tangible sign of interbreeding between Homo sapiens and Neanderthals, pushing their intimate contact far earlier than many had assumed. The claim is thrilling, but anatomy alone can mislead. As we revisit Skhul, we'll trace what the evidence really shows, what it could mean for our species' shared past, and why scientific caution matters as much as curiosity. Skhul was dug in the late 1920s with limited tools for fine-grained analysis. The new work revisits the first child's cranium and jaw using micro-CT, capturing internal structures without damaging the fossils. Researchers then built high-fidelity 3D models and placed anatomical landmarks to compare shapes across large reference samples of Homo sapiens and Neanderthals. This let them test, not guess, whether particular regions aligned with one lineage or the other, and to evaluate the pattern across the skull as a whole, crucial for spotting a true mosaic rather than a few isolated quirks. The overall cranial vault, its smooth, rounded curve around the brain, plots with modern humans. Deeper inside, the bony labyrinth of the inner ear clusters with Neanderthals. The lower jaw shows a stockier, more retromolar-friendly geometry. Tooth microstructure trends, Neanderthal-like, vascular impressions within the skull appear underdeveloped by modern standards. No single trait is definitive, but together they form a consistent pattern. Sapiens-like outer architecture paired with Neanderthal-like internal and mandibular signatures. Across Skhul burials, individuals display different mixes of these traits, a hallmark of mosaic morphology that the authors argue best fits prolonged contact and possibly interbreeding in the Levant. Taken together, the Skhul child's traits look less like a one-off anomaly and more like mosaic morphology, a patterned blend that, across other Skhul individuals, appears in shifting combinations. That pattern could fit admixture, but anatomy is a noisy proxy. Juveniles vary as they grow. Some characters are shaped by life history. Others can be retained from shared ancestors rather than acquired via hybridization. With only one juvenile examined in depth so far, the case is suggestive rather than definitive, a hypothesis waiting on biochemical proof. If borne out, the mosaic points to prolonged local interaction between modern humans and Neanderthals in the Levant, more than a brief contact window. It could also push interbreeding back by tens of thousands of years, forcing dispersal models to allow repeated encounters and gene flow this early and this far south. In that picture, Skhul becomes a demographic snapshot, overlapping populations exchanging mates, ideas, and perhaps technology. But the bar for rewriting timelines is high. Ancient DNA or another chemical marker will need to close the case. Modern humans and Neanderthals share an African ancestor. The lineages split greater than or equal to 500,000 years ago. Neanderthals appear in Eurasia by approximately 400,000 years. H. sapiens emerges in Africa approximately 300,000 years, with forays into the Levant before 100,000 years. The Levant, a corridor between Africa and Eurasia, saw recurrent occupations at caves such as Skhul and Kafse. In these narrow landscapes, small groups shifted with climate pulses, creating repeated windows for contact rather than a single migration. Ancient and modern DNA indicate our species and Neanderthals interbred much later, about 50,500 to 43,500 years ago, leaving 1 to 4% Neanderthal ancestry in most people with deep roots in Europe and Asia. Those signals reflect multiple introgression events and persist unevenly across our genomes. Fossils place both groups in the Levant earlier, but molecular clocks anchor major gene flow in the late Pleistocene. Skhul's anatomy challenges that timing. Only genetic or other biochemical evidence from the site can substantiate an older pulse. Anatomy is an indirect, noisy proxy for ancestry. Juvenile fossils complicate things because growth changes skull shape, tooth microstructure, and vascular imprints in ways that can mimic non-modern traits. Some features are plesiomorphic, retained from a shared ancestor, so they needn't signal hybridization at all. Taphonomic distortion, pressure, heat, mineral replacement, can subtly warp measurements, and small samples inflate apparent patterning. Comparative baselines matter too. If reference datasets underrepresent early H. sapiens variation or regional Neanderthal diversity, statistical assignments can drift. Finally, traits covary. A few correlated characters may look like a pattern without reflecting real gene flow. Decisive tests are biochemical. Best case, recoverable ancient DNA from the Skhul child or nearby burials, even short fragments, to quantify Neanderthal ancestry and date admixture. If DNA fails, paleoproteomics, enamel or bone collagen peptides can provide lineage signals. Sedimentary aDNA from the grave fill is another angle. Parallel steps strengthen the case. Direct, high-precision dating of the bones, micro-CT of additional Skhul individuals to see whether the mosaic repeats. Pre-registered morphometric protocols and blind reanalysis across labs, and integration with isotopic data, mobility, diet, to test scenarios of repeated contact. Short of genetics, only converging, replicated lines of evidence should move the timeline. Skhul isn't just bones, it's intention. Multiple individuals were deliberately interred in the same rocky outcrop, turning a natural ledge into a place with social meaning. That hints at memory and group identity, returning to the same spot, marking who belongs, and possibly coordinating rites around death. Such placemaking looks like an early form of territoriality, a claim to landscape anchored by ancestors. If true, it nudges complex social behavior, planning, ritual, symbolic thought, far earlier than the agricultural revolution, which is often credited with codifying territory and inheritance. If Skhul's mosaic reflects prolonged coexistence, daily life likely involved more than occasional encounters. Neighboring groups could have traded objects, shared raw materials or know-how, and formed mating ties that stitched small populations together. Burial practice itself may have traveled across group boundaries, evolving locally over generations. None of this requires large numbers. In Ice Age corridors like the Levant, repeated small contacts can reshape culture and, perhaps, genomes. Rescan remaining Skhul adults and juveniles with micro-CT using harmonized landmarks, pre-registered protocols, and open data. Attempt ultra-short fragment aDNA, single-stranded libraries, targeted capture. If DNA fails, turn to enamel proteomics and collagen peptide profiling. Secure direct ages via U-series/ESR on teeth and OSL on sediments. Add micromorphology to verify burial context and strontium/oxygen isotopes to track mobility. Compare systematically with nearby Levant sites, e.g. Kafse, Tabun. Integrate Skhul into models with multiple out-of-Africa pulses and repeated admixture in Levantine corridors. replace tidy replacement narratives with braided population streams. If early gene flow holds, modern human diversity looks increasingly patchwork, shaped by small local contacts that influenced identity, behavior, and resilience across late Pleistocene landscapes. Skhul invites a recalibration. Contact between sapiens and Neanderthals may have been older, longer, and more local than tidy timelines allow. But anatomy is only a prologue. Until DNA or other biomarkers weigh in, the Skhul child remains a compelling maybe, an ancient whisper asking science to listen harder and prove it. Thanks for watching. If this deep dive sparked your curiosity, hit subscribe and turn on notifications. See you in the next adventure.